Piwi interacting RNA. PRNA is the largest class of small non-coding RNA molecules expressed in animal cells. PRNAs form RNA protein complexes through interactions with Piwi proteins. These PRNA complexes have been linked to both epigenetic and post-transcriptional gene silencing of retrotransposons and other genetic elements in germline cells, particularly those in spermatogenesis. They are distinct from microRNA in size 26 to 31 nt rather than 21 to 24 nt. Lack of sequence conservation and increased complexity. It remains unclear how PRNAs are generated, but potential methods have been suggested, and it is certain the biogenesis pathway is distinct from mRNA and sRNA, while RNAs are a PRNA subspecies. Characteristics. PRNAs have been identified in both vertebrates and invertebrates, and although biogenesis and modes of action vary somewhat between species, a number of features are conserved. PRNAs have no clear secondary structural motifs. The length of a PRNA is, by definition, between 26 and 31 nucleotides, and the bias for a 5' uridine is common to PRNAs in both vertebrates and invertebrates. PRNAs in Canorhabditis elegans have a 5' monophosphate and a 3' modification that acts to block either the 2' or 3' oxygen, and this has also been confirmed to exist in Drosophila melanogaster, zebrafish, mice and rats. This 3' modification is a 2-O-methylation. The reason for this modification is not clear, but it has been suggested to increase PRNA stability. It is thought that there are many hundreds of thousands of different PRNA species found in mammals. Thus far, over 50,000 unique PRNA sequences have been discovered in mice and more than 13,000 in D. melanogaster. Location. PRNAs are found in clusters throughout the genome. These clusters may contain as few as 10 or up to many thousands of PRNAs and can vary in size from 1 to 100 kb. The detection and annotation of PRNA clusters in genomes based on bioinformatic methods became more and more sophisticated during the past years. While the clustering of PRNAs is highly conserved across species, the sequences are not. While D. melanogaster and vertebrate PRNAs have been located in areas lacking any protein-coding genes, PRNAs in C. elegans have been identified amidst protein-coding genes. In mammals, PRNAs are found both in testes and ovaries, although they only seem to be required in males. In invertebrates, PRNAs have been detected in both the male and female germlines. At the cellular level, PRNAs have been found within both nuclei and cytoplasm, suggesting that PRNA pathways may function in both of these areas and, therefore, may have multiple effects. Biogenesis. The biogenesis of PRNAs is not yet fully understood, although possible mechanisms have been proposed. PRNAs show a significant strand bias. That is, they are derived from one strand of DNA only, and this may indicate that they are the product of long single-stranded precursor molecules. A primary processing pathway is suggested to be the only pathway used to produce Paq-10 PRNAs. In this mechanism, PRNA precursors are transcribed, resulting in PRNAs with a tendency to target 5' uridines. Also proposed is a ping-pong mechanism wherein primary PRNAs recognize their complementary targets and cause the recruitment of Piwi proteins. This results in the cleavage of the transcript at a point 10 nucleotides from the 5' end of the primary PRNA, producing the secondary PRNA. These secondary PRNAs are targeted toward sequences that possess an adenine at the 10th position. Since the PRNA involved in the ping-pong cycle directs its attacks on transposon transcripts, the ping-pong cycle acts only at the level of transcription. One or both of these mechanisms may be acting in different species. C. elegans. For instance, does have PNAs, but does not appear to use the ping-pong mechanism at all. A significant number of PNAs identified in zebrafish and D. melanogaster contain adenine at their 10th position, and this has been interpreted as possible evidence of a conserved biosynthetic mechanism across species. Ping-pong signatures have been identified in very primitive animals such as sponges and cnidarians, pointing to the existence of the ping-pong cycle already in the early branches of metazoans. Ping-pong. The PNA ping-pong pathway was first proposed from studies in Drosophila where the PNA associated with the two cytoplasmic Piwi proteins, aubergine, orb, and argonaute3, AGO3, exhibited a high frequency of sequence complementarity over exactly 10 nucleotides at their 5' ends. This relationship is known as the ping-pong signature, and is also observed in associated PNA from MeW2 proteins isolated from mouse testes. The proposed function of ping-pong in Drosophila or in mouse remains to be understood, but a leading hypothesis is that the interaction between orb and AGO3 allows for a cyclic refinement of PNA that are best suited to target active transposon sequences. Orb PNA are primarily antisense to transposable element transcripts and are believed to be the main factor in targeting deleterious transcripts through complementarity. Conversely, AGO3 PNA sequences are predominantly of sense orientation to transposable element transcripts, and are derived from the product of orb cleavage of transposon mRNA. As such, AGO3 PNA lack the ability to target transposable element transcripts directly. Therefore, it was proposed that AGO3 PNA guide the production of PNA that are loaded into orb by targeting newly exported PNA cluster transcripts. Several lines of evidence support the effect of AGO3 on the production of orb PNA, in particular from examining the PNA repertoire in Drosophila ovaries that are mutant for AGO3, and the tudor domain protein chemokine. The molecular mechanism that underpins ping-pong likely involves several PNA pathway-associated factors. Kine was reported to coordinate the loading of AGO3 with PNA, in addition to interacting with both orb and AGO3. However, the tudor protein, CRIMPA, was also shown to interact with both orb and AGO3 through its tudor domains while also binding itself through its N-terminal CRIMPA domain. Specifically, CRIMPA interacts with AGO3 in its PNA unloaded state, while its interaction with orb is dependent on the symmetrical dimethylation of arginine residues in the N-terminal region of orb. In silk moth germ cells, it was proposed that Vata protein coordinates the ping-pong mechanism of silk moth orb, Piwi, and AGO3. It is likely that the mechanism of ping-pong is primarily coordinated by CRIMPA, but factors such as chemokine and Vata, in addition to other factors, have necessary functions in the ping-pong mechanism. PNA phasing. The Drosophila PNA pathway can be separated into two branches: the cytoplasmic branch consisting of orb and AGO3 operating the ping-pong mechanism, and the nuclear branch, pertaining to the co-transcriptional silencing of genomic loci by Piwi in the nucleus, through complementary strategies. Two studies show that orb and AGO3 target cleavage triggers the phased loading of PNA into Piwi. Phasing begins with the targeting and cleavage of a complementary target by either orb or AGO3, associated with a responder PNA. Once cleaved, the targeted transcript is then processed further by a mechanism believed to require the mitochondrial-associated endonuclease, zucchini, which leads to the loading of Piwi protein with sequential fragments of the targeted transcript. In this way, the orb or AGO3 responder PNA sequence cleaves a complementary target that is then sliced at periodic intervals of approximately 27 nucleotides that are sequentially loaded into Piwi protein. Once loaded with PNA, Piwi then enters the germ cell nucleus to co-transcriptionally silence nascent transcripts with complementarity to its PIRNA guide. It is currently unknown whether phasing occurs in other organisms. Function. The wide variation in PIRNA sequences and Piwi function over species contributes to the difficulty in establishing the functionality of PIRNAs. However, like other small RNAs, PIRNAs are thought to be involved in gene silencing, specifically the silencing of transposons. The majority of PIRNAs are antisense to transposon sequences, suggesting that transposons are the PIRNA target. In mammals, it appears that the activity of PIRNAs in transposon silencing is most important during the development of the embryo, and in both C. elegans and humans, PIRNAs are necessary for spermatogenesis. RNA silencing. PIRNA has a role in RNA silencing via the formation of an RNA-induced silencing complex. Risk. PIRNAs interact with Piwi proteins that are part of a family of proteins called the argonauties. These are active in the testes of mammals and are required for germ cell and stem cell development. In invertebrates, 3 Piwi subfamily proteins, MIWI, MIWI2 and MILI, have been found to be essential for spermatogenesis in mice. PIRNAs direct the Piwi proteins to their transposon targets. A decrease or absence of PIWI gene expression is correlated with an increased expression of transposons. Transposons have a high potential to cause deleterious effect on the host, and, in fact, mutations in PIRNA pathways are found to reduce fertility in D. melanogaster. Further, it is thought that PIRNA, an endogenous small interfering RNA, endosona, may have comparable and even redundant functionality in transposon control in mammalian oocytes. PIRNAs appear to affect particular methyltransferases that perform the methylations which are required to recognize and silence transposons. But this relationship is not well understood. Epigenetic effects. PIRNAs can be transmitted maternally, and based on research in D. melanogaster, PIRNAs may be involved in maternally derived epigenetic effects. The activity of specific PIRNAs in the epigenetic process also requires interactions between Piwi proteins and HP1A, as well as other factors. Accessory proteins of the PIRNA pathway. Genetic screens examining fertility defects identified a number of proteins that are not Piwi-clade argonauties, yet produce the same sterility phenotypes as Piwi mutants. Drosophila pseudodomain proteins. Many factors required for the PIRNA pathway in Drosophila contain pseudodomains that are known to bind symmetrically dimethylated arginine residues, SDMA, present in methylation motifs of Piwi proteins. Piwi proteins are symmetrically dimethylated by the PRMT5 methylosome complex, consisting of VALWA, MEP50, and capsuline, DART5, PRMT5, TUDA, TUD, KEEN, KUMO, SPINDLE, SPNE, CRIMPA, TECUS, TAGE, VRITENO, VRET, PAPI, YB, FS1, YB, brother of YB, boy B, sister of YB, SOYB, non-TUDA Drosophila PIRNA pathway proteins. VASA, maelstrom, male, Drosophila nuclear PIRNA pathway proteins. Rhino, HP1D, deadlock, cutoff, SETDB1, Eggless, SUVA 3-9, investigation. Major advances in the PIRNA field have been achieved thanks to the use of next-generation sequencing techniques, such as Solexa 454 and Illumina platform sequencing. These techniques allow analysis of highly complex and heterogeneous RNA populations like PIRNAs. Due to their small size, expression and amplification of small RNAs can be challenging. so specialized PCR-based methods have been developed in response to this difficulty. References. Further reading. Lau, N. C. 2006. Characterization of the Pirna complex from rat testes. Science. 313. 5785. 363 to 367. DOI 10.1126. Science 1130164. ISSN 0036-8075. Kim, V. N. 2006. Small RNAs just got bigger. Piwi interacting RNAs. Pirnas. In mammalian testes. Genes Dev. 20. 1993–1997. DOI 10.1101. GAD 1,456,106. PMID 16,882,976. Gerard, Angelique, Sakadanandam, Ravi, Hannan, Gregory J. Carmel, Michelle A. 2006. A germline-specific class of small RNAs binds mammalian Piwi proteins. Nature. 442. 199–202. DOI 10.1038. Nature 04917. ISSN 0028-0836. PMID 16,751,776. Grivner, S. T. 2006. A novel class of small RNAs in mouse spermatogenic cells. Genes and Development. 20. 13. 1709–1714. DOI 10.1101. GAD 1,434,406. ISSN 0890-9369. Watanabe, T. 2006. Identification and characterization of two novel classes of small RNAs in the mouse germline. Retrotransposon-derived siRNAs in oocytes and germline small RNAs in testes. Genes and Development. 20. 13. 1732–1743. DOI 10.1101. GAD 1,425,706. ISSN 0890-9369. Carmel, Michelle A. Gerard, Angelique, Van der Kant, Henk J. G. Orcus, Deborah, Bester, Timothy H. Deruai J. Dirk G. Hannan, Gregory J. 2007. MIWI2 is essential for spermatogenesis and repression of transposons in the mouse male germline. Developmental cell. 12. 4. 503–514. DOI 10.1016. J. Dev cell. 2007.03.001. ISSN 1534–5807. External links. Ping Pong Pro, a software for finding ping pong signatures and ping pong cycle activity. Pirna Bank, a web resource on classified and clustered Pirnas. Protrack, a software for probabilistic Pirna cluster detection, visualization and analysis. Pirna cluster database.